生命医学领域是人工智能技术的重要应用场景，也是目前大语言模型应用前景最为清晰的领域之一。我们邀请将探讨如何将大模型技术应用于生物医学研究，如基因组学、蛋白质结构预测和药物发现等。今天下午我们邀请到了 Jure Leskovec等学者分享他们在生物医学大模型应用中的实践经验和创新思路，讨论如何结合生物医学领域的特点，设计性能更加优秀的模型与算法，提升模型在真实和未见场景下的适应性，并深入探讨大模型技术在生物医学领域的伦理和社会影响。首先让我们有请今天下午的这个 keynote speaker 来，他将会进行基础模型在全科医学人工智能中应用潜力的报告。让我们现在先把我们的麦克风交给吴泰玲，然后他会是这个 session 的 moderator.
It's my great honor to introduce Professor Jure Leskovec. So Jure Leskovec is a professor of computer science at Stanford University. He is affiliated with Stanford AI Lab, Machine Learning Group, and the Center for Research on Foundation Models. In the past, he served as chief scientist at Pinterest and was an investigator at Chan Zuckerberg BioHub. Most recently, he co-founded the machine learning startup Kumo.ai. Leskovec pioneered the field of graph neural networks and co-authored PyG, the most widely used graph neural network library. Research from his group has been used by many countries to fight COVID-19 pandemic and has been incorporated into products at Facebook, Pinterest, Uber, YouTube, Amazon, and more. His research received several awards, including Microsoft Research Faculty Fellowship in 2011, Okawa Research Award in 2012, Alfred Sloan Fellowship in 2012, Lagrange Prize in 2015, and ICDM Research Contributions Award in 2019. His research contributions have spanned social networks, data mining and machine learning, and computational biomedicine, with the focus on drug discovery. His work has won 12 Best Paper Awards and 5 10-year Test of Time Awards at premier venues in these research areas. And one more important thing is that Jure is also my advisor during my postdoc, and it's my great privilege to be advised by him. Now the floor is yours, Jure.
Yeah, thank you very much for this nice and kind introduction, Tailin. Great to be among all of you and excited to talk to you. I would appreciate if I could get screen sharing permissions so I could show you my slides. Great. Everyone can see my slides?
Yes.
Super. So I'm, as I said, very excited to be able to talk to you all today. And I prepared a talk about foundation models, and in particular their application to sciences. And this is really a joint work, as you are going to see, with a number of people, students, postdocs, and other collaborators, professors here at Stanford University, in multiple departments, medical school, biology, and so on. Right? We are all right now in this big language model revolution where we basically take a lot of natural language text, and then the transformer architecture is being used to train a language model to predict the next word, the next token across these huge amounts of data. And these large-scale pre-trained models can be adapted, fine-tuned, or via in-context learning, so via prompting, to many different applications, from question answering, text classification, information retrieval, and so on and so forth. And we are all very excited about this breakthrough. And what is amazing about these language models is that they can do a lot of different things. They can summarize a given paragraph of text. They can, they understand emojis and can formulate interesting responses. And these types of models have been extended also to multimodal domains, meaning that they can process both images and text. And they're able to describe images as well as and generate text about them, or you can give them a text and they are going to generate you realistic looking images from, you know, teddy bear on a skateboard in the New York, or the Golden Gate Bridge actually being a train bridge with a Caltrain riding over it. And another domain where these language models have been amazingly successful and blew everyone away is in terms of code. So basically understanding the computer code and being able to translate between the textual description here and the computer code to execute this textual description at the bottom. And a premier example of this is GitHub Copilot. But these types of models have made a big impact elsewhere as well. In particular, one of the biggest scientific breakthroughs of the last few years is the Google's AlphaFold that takes as an input the amino acid sequence of a protein and then is able to perform protein folding. Basically, it's able to predict or estimate the spatial structure of that protein based on its sequence. So based on these amazing advancements in AI and kind of powered by large language models, graph neural networks, and so on, we at Stanford kind of try to unify all this into this notion of foundation model. And we call that a foundation model is a model that is trained on a broad data. So basically, it's a big model trained in a self-supervised way at a large scale, and is then adapted basically lightly and effectively to a wide range of downstream tasks, right? So to us, this notion of foundation model is much more general than just the language model. And why is important about this notion of foundation model is that it really brings the paradigm shift into AI, right? If we think of, for example, the old world of machine learning, where really the question was, how do we do prediction? And it was all about learning algorithms. Right? How do we build learning algorithms? In the deep learning era, it is all about representation. It's about the feature. So it's like, what is a good neural network architecture that can learn a good representation? But now, in the age of foundation models, it's actually a step higher in the hierarchy. It's about functionality. It's about what is the ability of this model. And it's really, we talk about models. We don't talk about algorithms. We don't talk about architectures, but we talk about the abilities of these particular models. So at Stanford, we established a Center for Research on Foundation Models that is unique interdisciplinary team of 30 plus faculty, 200 students and postdocs spanning across all kinds of departments, from computing to healthcare, education, law, economics, philosophy, as well as humanities and sciences. And our mission is to make foundation models more transparent and more accessible. We have authored a large paper on the opportunities and risks of foundation models that you can find on arXiv. Where we put together a comprehensive list of opportunities, capabilities, applications, technologies, as well as societal challenges in adoption, implementation, and deployment of these large-scale foundation models in many different domains. So in this talk, what I plan to talk about is several different aspects around the efficiency of this type of foundation models. I'll talk about some technical advances. I'll talk about applications in biomedicine, health, and I'm also going to talk a bit about social responsibility and how do we really deploy these large-scale models to societal benefit, to very important societal applications. So first topic, my talk will kind of be structured in a few different parts. And in the first part, I want to really talk about foundation models for biological sciences, in particular for molecular biology and drug discovery. And, you know, this is joint work with Yanai, Maria, Yusuf, Keshin, and Camilo. in particular, our goal, right, if a large-scale language model is like it's like a map of human language and human knowledge, we would like to build a map of biology, right? We would like to build a model that really understands and can make predictions about human biology. And this is extremely important for medicine, for drug discovery, understanding fundamental of human biology. The question that is interesting first to ask, if you say, if I want to go build a foundation model for biology, the question is, how do I do this? And we really need to think about, what is the foundation model for? What are the building blocks that make that domain? And do we have a large-scale data set available? Right. So, for example, you can say that for language, we have word sequences, and there's an entire internet of them. For vision, images are pixel arrays, matrices of pixels. We have a lot of data again on the internet. And for example, for proteins, we have an amino acid sequences, and there's a lot of these protein sequences that we can use to train large-scale, let's say, protein models. Those are molecular. Those are kind of too low level. So the question is, what is the right granularity to really model life, to model biology? And what we argue is that the building block of life is a cell, right? So we want to use cells as building blocks of life. And the question is, do we have a large-scale data available that allows us to build a foundation model of biology of cells? And in this case, the question is, how do we characterize cellular behavior, right? And the way, like, if there is a quick slide about biology, it's really that we have our genotype, we have our DNA, our genes, that then through this transcription process get transcribed into RNA, which then gets translated into proteins, right? So we kind of go from this genotype through the RNA that then is translated into proteins, and proteins lead to the phenotype, lead to the actual instantiation of a given being. So what we would really like to understand and capture is this process, and is in a sense what phenotype is defined by is this proteins, this RNA, this DNA that defines and makes life. So we have decided to work at the RNA level, because it's a building block between having the genotype and then that being translated and transcribed into the proteins that then regulate cellular functions. And there is a lot of different proteins in human bodies, right? And the reason why RNA is a good idea is because RNA molecule is the transcript of the information contained in a gene. And expression of a gene, the activity of a gene, is measured by the RNA count, by the amount of these RNA molecules. And the last thing to say is that the expression of all genes in a cell can determines the identity and function of the cell. So if we are able to capture how much RNA is in the cell, then we are able to measure the expression level of a gene, and really that regulates biological function. So basically the identity of the cell, the function of the cell, the state of the cell, its identity and function can be determined by these RNA counts. So maybe one way to think of this is the following, right? If we think there are several different ways how DNA, RNA sequencings have been done in the past. The old school method is what is called bulk RNA sequencing, or that is, for example, used in genome-wide association studies, where basically it's like a smoothie, right? You take all the cells in a sample, you mix them together, and out you get the RNA. The RNA counts across all the cells in a given sample. So it's like mixing all the fruit together. Then the next innovation was what is called the single cell RNA sequencing, where basically for every single cell you can sequence its RNA. So you can capture the state of every cell. And the way we can think of this is that now the fruit is not mixed, but we kind of know how much of pineapple, how much of strawberry, how much of kiwi is in my fruit salad. And the last kind of the latest technology in this space is called spatial transcriptomics, where basically not only we know the RNA counts of the individual cells, but we also know how the cells are positioned in space and adjacent to each other. And this is called spatial transcriptomics. And the biology is kind of moved from the left very far to the right, where now we can understand what are the cells and how they are spatially organized. And the reason why do we want to do this profiling of the cell behavior is because all cells in your body have some DNA, have the same DNA. You know, the cell on my skin has the same DNA as the cell in my heart. So, you know, how do cells carry different functions? If they all have the same DNA, why are they not all the same, right? But I have different tissues, I have different organs, I have brain, I have lung, but I have all the same DNA. So that's the first question. And then the second fundamental question is, you know, how do the cells change during disease or during aging? Because if we understand this state, then we might be able to take a sick cell and transform it into a healthy cell, for example. So single cell RNA sequencing can provide precise measure of characteristics of individual cells and allows us to answer these very important questions. So if we think about how is this data represented, it is represented in these big matrices where every column is a different gene. We have about 20,000 protein coding genes. So about 20,000 genes get transcribed into RNA that then gets translated into the proteins. And for every cell, for every different cell, we have the count of the RNA for that particular gene. So that's basically the input. It's like a 20,000 columns and as many cells as you can get. And in collaboration with Chen Zuckerberg Initiative, this is a organization founded by Mark Zuckerberg and his wife, so basically the founder of Facebook, to discover the mechanisms of human health. And they have released the largest data set of human cells that includes 40 million different cells across many different cell types, across many different human tissues. So basically we have now a matrix of 20,000 columns and 40 million rows, right? And that is amazingly interesting. This is the large-scale data we can use to train our models. There are two big challenges when we wanna build, let's say, a model of biology using this type of data. The first thing is that a lot of prior work treats this gene to cell matrix simply as an index in the matrix, right? And includes no information about the gene sequence or what the protein that a given gene codes for is. And when traditional algorithms process this data, they have no knowledge of genetic variants and chromosome locations. That's the first challenge. And then the second challenge is that if we take these matrices, let's say, generated by different groups across the world, and we look at these RNA counts and maybe try to project this in the common space, basically we find out that all these data sets are kind of completely different from each other. So even if we all take, you know, sample from the same human, and these data sets should kind of overlap in this, let's call it embedding space, in this low dimensional space, basically the experimental artifacts are much, much bigger than the amount of biological signal. So basically all today's methods are able to learn is that data comes from different data sets. But it's not that, you know, all the lung cells from all the different humans would map on top of each other, which is what we would like and what we'd like to do. So the way we can resolve this is to say, you know, how can we enrich our data to encode more than just this gene expression, just this RNA count. And we have developed this notion of what we call functional expression that transforms expression to reflect biological function rather than specific gene. And the way we do this is to use large-scale protein language models, where we can now take the sequence of the protein, embed that sequence of the protein as a characterization of the function and the role of the gene. So this means that we can now take very different species that have very different genes and very different proteins and embed them into the same space, and we can start reasoning about the relationships between different species. This provides a huge improvement over what has been possible before. We are in particular using large-scale protein language models developed by the team at Facebook, in particular ESM, to encode the whole protein universe. This was trained on over 250 million protein sequences, and it has been shown that these protein embeddings encode both structure, molecular properties, as well as the orthology information. And we have developed a method that we call Saturn, and the preprint is available on arXiv, listed down here, where basically we can take these RNA sequence counts from many different organisms, maybe from human, from mouse, from lemur, and then we take also their protein sequences that are coded by each of these different genes, and use the large-scale protein language model to allow us to reason how do these different genes across different organisms, how do they relate to each other, to this concept of a macrogene. And the concept of macrogene is a gene that is kind of is an artificial gene that kind of connects all the genes in the real world. And through this embedding, we are now able to take any cell of any organism and embed it into the common embedding space. And from there, we can, for example, solve for many different tasks. We can create multi-species cell atlases. We can identify differentially expressed macrogenes and really reason about the underlying biology. We can transfer cell type labels across species. We can, re-annotate cell types across species. We can identify protein differences and so on. And we have actually found out that our method really allows us to accurately transfer cell type annotations from model species to new species. So here we would train our model on, for example, use the zebrafish as a model organism, and we would then take a different organism, frog, and try to annotate into different cell types the cells of the frog. And we can see that we are able to do this 120% better than previous methods like SAMAP, Harmony, SCVI, or Scanorama. So a huge boost from this foundation model view and using embeddings of protein sequences to be able to do this annotation. And I'll just show you something that is truly impressive. We are the first method that can take cells from 5 different organisms, here colored in different colors, where every cell here, every dot is a different cell. So here we are taking cells from all these organisms and we map them into the joint space. And what you see is that we find clusters, but what is important is that these clusters are, the colors in the cluster are mixed. What this means is that basically the cells of the similar type, regardless of what organism they come from, they map to the same cluster. So for example, this cluster down here is all the B cells. So it's human B cells, macaques B cells, mouses B cells, and so on, right? And here is a cluster of macrophage cells, and right, you see it's how it's mixed by the color in terms of what organism it comes from, but it's very clean in terms of what is the cell type. So this is now the first time when we can take data from very different organisms and map them all to the common space. So this is the first thing I want to talk about. This is like building this cell space in which cells from any organism can be mapped, so we can understand the state of those cells, their identity, their function, and so on. Once we understand that, we don't only stop there. It's not only about identifying, but also about predicting behavior of the cells so that we can engineer cells that we would like. And this becomes very important because I can say, I have a sick cell, how do I make it healthy? And the way we can think of this is that basically we can think of this as a predictive task of how do we predict the outcome of a cell perturbation, where basically I can say, I have a cell in a given state, here a gene expression state, And then I can say, what if I suppress or amplify, what if I perturb, gene 2 and gene 4? What is going to happen to the state of the cell? Right? Maybe I take a drug that inhibits the work of proteins coded for by gene 2 and gene 4. What happens to the state of the cell? What happens to the expression levels of genes in that cell? Right? So the question that we want to answer is, what is the gene expression response of perturbing a combination of genes, and what is the resulting cell? And we have built an approach called GEARS that was just published in Nature Biotechnology. The paper is cited down here, where we built a deep learning model constrained by prior knowledge using knowledge graphs of genetic relationships, as well as a knowledge graph of understanding perturbation relationship graph through the gene ontology, that allows us to basically take in as an input the state of the cell, so basically the state of the unperturbed cell, the information, what genes in the cell we'd like to perturb, and then through this neural network transformation, we predict what is the outcome of the perturbation. We predict what is the resulting gene expression of the perturbed cell. And what is interesting in this case is that we can make very accurate predictions, and that actually the underlying process is very complex. What I try to show you here, for example, is if I take a cell and I perturb two genes, here they are listed. And now I'm asking, what is the effect of the perturbation of that cell on the rest of the genes? So here, every three, I have different genes, and the legend here is the following. The gray line is what is the result expression of the gene after these two genes were perturbed. So here, the expression of this gene increased, the expression of this gene increased a lot, while, for example, expression of these genes actually decreased. Red is our prediction. And you can see how our prediction, for example, matches the truth quite well. The yellow is prediction from a linear model, where we basically say, what is the outcome if we predict the first gene? What is the outcome if we predict the second gene? Let's add the two up and see the outcome. And you see how the yellow is actually either severely over-predicting or severely under-predicting in many different cases, which means that this underlying process is heavily non-linear. And we find that GEARS, our method, has 50% higher precision in detecting the correct nonlinear genetic interactions, and it's twice as accurate in predicting the strongest interactions. So you can say, why is this useful? This is amazingly useful capability because it helps us in drug target discovery. You can say, I have a healthy cell, what genes should I target to make it look I have an unhealthy sick cell, what genes should I target to make it healthy? So it allows us to identify therapeutic targets that can reverse disease phenotypes. It also allows us to computationally identify new genetic interactions, predict genetic interactions. And it's amazingly useful for cell re-engineering, where we can say, how do I take a cell in one state, maybe an old immune cell, and how do I make it look young? So this allows us to enhance precision and efficacy of cell reprogramming by computationally identifying the targets. So to summarize this part of the work, I wanted to say the following. Biology is not a language and is not natural language, and care needs to be taken when adapting tools from natural language or vision onto biology. And by building foundational models that leverage biological data and biological kind of units, tokens, we can build zero-shot tools for understanding cell biology and really contribute to a revolution in drug discovery, healthcare, and our understanding of life. Currently, we are working on on exciting problems around exploring the structure of the embedding space to discover new insights about cellular hierarchies and functions. And we are also incorporating patient-derived single-cell data to learn clinically relevant information, especially around personalized medicine. So that's the first part of my talk. In the second part, I want to touch on two things. First, I really want to talk about our paper that we recently published in Nature that with Michael, who was the lead author, and then Michi and Hamed, about foundation models for generalist medical artificial intelligence. Really thinking about how do we think about foundation models for medicine. And the problem is that medical data is often scarce and very heterogeneous, right? We have text, we have electronic health records, we have measurements, we have EKG, we have images, and so on and so forth. And also chemical prediction tasks are highly complex and require a lot of domain knowledge. And, meanwhile, prior medical knowledge is hardly used in current medical AI systems because most of the medical knowledge is actually written in books and in ontologies and so on. So our insight is that medical foundation models with structural medical knowledge unlock generalization capabilities and enable applications across many medical prediction tasks. And while conventional approach to medical AI is to curate some data set together with some labels and then build a prediction model, we want to kind of step away from this because there are many drawbacks of this approach. These models are very narrow for only specific tasks. Manual data labeling is very costly, and large task-specific data sets are required for good performance. The question is, what's the alternative? And we think that pre-trained models are the alternative, right? Pre-trained models have broad applicability. They are trained in a self-supervised way, so they don't require any labels, and they don't require large task-specific data sets, but they can be trained on large task-agnostic data sets. So we have proposed this notion of generalist medical AI, which is basically a multimodal foundation model that can be trained on many different types of data, from images, EHR signals, as well as this what is called omics data, so genomics and transcriptomics and so on, as well as being able to ingest medical domain knowledge in terms of publications, textbooks, knowledge graphs, and clinical nodes, and then have the zero-shot capability so that they are able, on just a few examples, learn to do a task they have never seen before. The benefits of this is that basically we get flexible multimodality, that we are freely combine and interleave different data modalities. We allow for dynamic task specification, where we solve tasks on the fly based on the instructions, so basically zero shot or few shot kind of in context type learning. And these models have a lot of knowledge because they allow us to reason with structured domain knowledge. And there are many different use cases for these types of models, from bedside decision support to generating grounded, let's say, reports, as well as serving like as a co-pilot for doctors when they are deciding on medical procedures. There is, of course, also a lot of challenges in these areas around validation, verification, social issues, biases, as well as patient privacy that is very, very important. But the technical challenge that needs to be solved to give rise to these generalist medical foundation models is really the challenge of multimodality, right? Where we have the, maybe the MRI scan of the image, we have the definition or the description of what happened to the patient, and then we need a prediction of, what is the risk of bleeding, of brain bleeding in this case. And the problem with current multimodal models is that basically they lack knowledge and they hallucinate. And I just give you an example with this challenge is that, for example, if you ask a multimodal foundation model today, to generate, what does an Armenian church look like? It is going to give you a picture of a church, but it doesn't look right. Armenian churches have this type of style. But if you ask, what is the name of this place, here it is going to say it's Dragon, Tiger, Pagodas next to the fireworks, but really but what the model is saying, it's saying it's in Empire State Building and fireworks, but these are really Dragon and Tiger pagodas with the fireworks. So our inspiration how to solve for this type of hallucinations and how to bring more information, more domain knowledge into these large pre-trained models is through retrieval augmented language models, where, for example, for unimodal, so meaning just text-based language models, so basically we can use Wikipedia and retrieve from it and then use that as a part of the prompt in the generator, into the language model to give an accurate answer, right? So we would say, who is the president of the United States? We would retrieve knowledge from Wikipedia and then use that as a context to the language model generator to provide the answer. We have developed, together in collaboration with Facebook, one of the first retrieval augmented multimodal model. Basically, it's a retrieval augmented model that allows for reasoning across multiple domains. So here, for example, we work with language and image domain, where we can get an image of saying, generate me an image of a Labrador sitting on a bench near water. We would retrieve from the multimodal memory. Maybe what we retrieve is a Labrador retrieving sitting on a bench, and we use this as a context for our multimodal model generator to now generate the image of a Labrador next to the water. And here is the paper that we have published at ICML this year about RACM3 is the name of our model. And our innovation is to build this multimodal retriever where we have a dense retriever with mixed model encoder, where we take basically a score of the query, the text, and what we retrieve from the memory to produce the score. And our score is basically computed by basically having this mixed model encoder that takes the query, either which is image or text, or the mixture of the two. And then the same encoder can be applied to our knowledge base to encode, and we can retrieve this based on the score, based on the cosine similarity. And here, our mixed model encoder is actually an extension of CLIP, where you have a CLIP text encoder, CLIP image encoder, and then we do the mean pooling between the two. We have the way basically this works is that the way the generator works is that basically we retrieve examples. This would be image text pairs, and we use these retrieved documents as context, and then we insert the main document, and then ask for the model to generate the response. And I can show you that this actually works really well. For example, if you ask our model, generate French flag waving on the moon's surface, we would retrieve the image of the French flag, and the output is actually the French flag on the moon's surface. If you ask stable diffusion or vanilla CM3 model without retrieval, it actually generates American flag on the moon's surface. Or if you say we want an Oriental Pearl Tower from Shanghai in oil painting, if you ask prior models, they generate something that's an oil painting, but clearly the building is not the right one. While the retrieval-based model would actually retrieve the image of the Oriental Pearl Tower and now provide it as a context so that the correct image can be generated. And this way, the results get much improved. So now going back to my initial example of an Armenian church during a sunny day, our model actually retrieves the picture of Armenian church and use it as a context to generate realistic outputs, while, for example, the baseline models don't do the right things. Or if you say photo of the Statue of Liberty standing next to the Washington Monument, we can retrieve Statue of Liberty and Washington Monument, and the model is going to do the right generation. While if you just directly ask prior models, it is not generating good results. So with this innovation, that inspired us to build the first multimodal medical model that has zero-shot capability, and we call our model MetFlamingo. And there is a paper on arXiv, as well as Hugging Face and code on GitHub. But basically, what we use as a backbone language model, we use the Llama 7 billion language model, and then we also use a vision model. And to fuse them together, we are fusing them through the Flamingo fusion approach. And then we are training and further training or fine-tuning this pre-training on multimodal medical literature, where we have both text and images. And we used paired interleaved pre-training to basically build this multimodal, to allow for multimodal in-context learning. And the question, once we train this type of models for medicine, is how do we evaluate them? And the problem is that the medical visual question answering systems are very simplified. They don't ask a question, what is the under the diaphragm? And here we would have to answer, it's free air. But really, the way they work is they actually give you several possible answers, and the machine chooses the right one. So actually, in this case, in one of the most popular data sets, here are the possible answers to choose from, and there is a correct one and two completely incorrect ones that have nothing to do with the question. They are not even plausible. So it's kind of too easy to do. So what we have actually been working on now, and what we have developed, is a tool for human evaluation of medical foundation models, where we can basically start with an image, with a question, and then we say, here is the correct answer, and then we say, okay, what's the prediction of the model? How good is this? And how even plausible are other answers? And this way, we can get much more trustworthy evaluation of these important models. And because our model is has generation capability and has a few shot capability, it actually allows us to generate rationales. We can start with an image, ask a question about it, and our model is actually going to provide the proper answer. Because we allow for multimodal few shot prompts the way we train our model. So it has this few shot ability. Here I'm showing you, for example, two examples where we have the context, we have the image, and we have the question. And for example, if you would go ask GPT-4 for the answer, it actually gives you the wrong diagnosis. And it's partially correct because it's talking about left wing of the blood of the lungs, but the diagnosis is wrong. While the MetFlamingo actually gives the correct diagnosis also and correct pathology, and notices that it's the left-sided issue, not the right-sided issue. Here's another example where we basically have a microscopic image of cancer. And of course, models don't see this caption. Model just sees the patient description, the image, and the question is what represents the most significant risk factor for this patient's condition. Again, the correct answer is smoking, and our model does well. While the GPT style model would completely fail. So that's the second part I wanted to talk about, which is really about the importance of multimodality and how multimodality can really lead to use of foundation model for medicine, for medical sciences, and can help us improve human healthcare. The challenge in that is in evaluation. So I showed you some examples and some ideas how this can be overcome. So the last thing I want to talk to you about today is about foundation models as research agents. And this is really work with Qian, who's a PhD student co-advised between me and Percy Liang. And the way to motivate this work is that capabilities of large foundation models have really risen over the last few years, from handwriting recognition, speech recognition, image recognition, language understanding, reading comprehension, and so on and so forth. And all of these are kind of at the superhuman performance. And we have also seen these capabilities of large foundation models across many different tasks, from writing code to molecules to captioning to images and so on and so forth. And if you think about what are these large language models trained on, they're basically trained on the entire internet, which also means they have read all the research papers that are out there on the internet. So it means that these models, in a sense, have vast scientific knowledge, but also they have the ability to reason and they have an ability to plan. So what does this mean is that, could we build research agents that are LLM based? So basically, could we build an AI-based research agent or research assistant? And this is really the project on which we started at Stanford. So our work is that we are going to soon release what we call an ML agent benchmark, which is the first benchmark for evaluating AI research agents, where we provide a set of well-scoped machine learning tasks, basically a data set and a description of the machine learning task, where, for example, we say, here's the data set, and the success is to improve the model performance by 10% on the data set. And the idea is to build an automatic agent that is going through iteration, build a model that achieves this performance. And we have actually built a prototype LLM-based AI research agent that can use the sheer amount of prior knowledge on the internet in the LLM to help the research process and innovate and find the model that works well on a given data set. So the way our ML agent works is that tasks is specified by the research problem definition and by a workspace that includes the data set, the files, and so on. And this is where the AI agent starts. And then this agent first, basically, what usually it will do, it will first say, let's read the files. It will read the files and it will decide, oh, this is an image classification problem. Let's implement a convolutional neural network. And it is actually going to output code, in this case in Python, to be executed, right? It is going to then basically edit this code and provide a diff that then can be pushed into GitHub. And then the model can also, the agent, can also then execute this code to see how well the code works on the underlying data set. As it gets the output from executing the training job, then the agent examines the results. We ask it to self-reflect, and then it suggests revisions and outputs another diff, so basically another edit to the code that then can be again rerun. The results are then of that run are again ingested into the agent, which then decides what is the next step. So this is really a general framework for specifying research tasks, and we provide agent a file system and compute cluster access as actions. And then we record the interaction trace, basically these actions and snapshots of what the agent is doing to automatically build this model that performs well on a given novel machine learning task. And we automatically evaluate the research agent. What's the quality of the final model, as well as what is the process and efficiency that the model took to come to the answer. So the way we would do this is through the prompt, where we would say, you are a help research assistant, you have access to the following tools. Here is your research problem for you. And then we say, here's a summary of relevant actions, decide what you want to do. And then the agent would take the action and say, oh, I want to reflect, I can plan my research, here is what I'm thinking. Then it's takes the action, which is, okay, let's execute the script, and provides the parameters of that action. So it would be like, let's execute train.py. The train.py gets then executed, and the output is then put into the research log so that the LLM can consume this output and then decide what it wants to do next. So the prompt leads to action. Action executes a tool that generates an observation that gets put into the research log, and then the agent can then retrieve from the research log to decide on the next action that then gets executed. So our approach to building this agent is this notion of think and plan before act. So we ask our agent, our assistant, to reflect, plan out what it wants to do, and then we ask it to act, which is basically output what is that you want to do next. In this case, it would be, okay, execute my training script. And we have curated a number of different data sets, like from CIFAR10 to IMDb to many others, where basically the agent just gets the data set and the description of the machine learning problem, and it has to generate the code that solves that machine learning problem. And here we are comparing GPT-4 in dark red as a research agent with GPT-4 without retrieval. Claude, these are large language models from Anthropic, as well as AutoGPT and LangChain. And what is interesting, you see that for quite a few of these tasks, today's language models and our baseline prototype implementation of the research agent actually allows about 50% success rate on some of these data sets. So seems that it is plausible to build this type of AI research assistants. So what we also do, we find that current approach, our baseline implementation, makes quite a bit of errors. And when we classify the errors across different language models that are used as a base for our agent, we see that, for example, Claude from Anthropic tends to hallucinate more than GPT-4. It is worse at debugging, but it is better at planning than GPT-4. GPT-4 seems to be not good at coming up with a good plan and reaching success at the end. So GPT-4-based research agent avoids hallucination and being stuck with debugging, but it tends to fail more due to bad plans that do not help to make progress. So what I wanted to say here in this part is this interesting idea of, can we use large-scale language models to really build research assistants? So basically AI-based research assistant agent. And at Stanford, we are now building the first benchmark and the first baseline implementation of this, so that the rest of the community can take our work and build better agents and see, can we achieve 100% success across all these different machine learning data sets and across all these different tasks. So that's the exciting part here. So to quickly conclude, the Center for Foundation Model Research at Stanford is a platform for training domain-specific foundation models for health, biomedicine, and also focuses on technical advances like new architectures, optimizers. It turns important technical advances into applications. I showed you, for example, retrieval-based multimodal models. And we also develop technical advances driven by societal responsibility, for example, focusing on privacy and evaluation, which is very important for the medical domain. And we anchor social responsibility, the norms, in applications of medicine as well as biology. So with this, this concludes my talk. I would like to acknowledge all the students of my group, as well as postdocs, here's Tylin. Also, thank you for all the agencies and our industrial partners that make our research possible, as well as our collaborators. So thank you so much, and yeah, very happy to take a question or two.
Yeah, thank you very much for the wonderful talk. First, let's have the honored guest to have some questions.
Yes, so Jure, thank you so much for the talk. It's great, and thank you for staying so late for us. I saw that you were already yawning a little bit. I have a question about the joint training for the multimodal data set. When you do the kind of like the cross training, you know, when you say the representation from both sides, both the multimodal side as well as the textual side of it, what is the kind of like the right attention mechanism to connect these two pieces, and how do you account for the training for that? So that's my kind of like first question. And second question is that there are other ways to do this multimodal type of work, right? A lot of them are actually using, like my own work is using more like a neurosymbolic approach, where you have a domain-specific language to create these plans, and then use this plan to generate functions, you know, like using plugins type of stuff, and then do like, you know, you see the Allen Institute's work on the BSProg, right, or the other tools like that are using similar approach. What do you see in between this approach and also like your approach, especially the one that are using information retrieval to create these you know, these agents or these multimodal capabilities?
Yes. Thank you for a very good set of questions. In terms of multimodal, we really think that kind of flamingo type approach is the right way to go. It's kind of the heaviest, it's the hardest to do, but we think is kind of the right one, and it's the most expressive one. So you know, rather than trying to tokenize everything into some language, we think that this kind of, you know, pre-trained language, pre-trained vision, and then the cross in the flamingo style is the way to go. And we think it's better than, for example, this type of clip type models. Now for the multimodal retriever, in that one, as you saw, the importance of being able to do this kind of multimodal retrieval to have a trusted knowledge base from which you can retrieve and provide context, almost like a reminder to the language model, to the underlying model, is very important. We think that this retrieval capability is one of the kind of very fundamental ways about how can we prevent hallucinations and how do we make these language models or multimodal models work better. An alternative approach would be to do fine-tuning, but then fine-tuning can be only done either on a specific data set or on a specific task, and it's unclear whether the model really learned that particular, let's say, private data or that knowledge. So we find we have kind of more trust in the retrieval-based approaches, and we are very excited about this type of multimodal retrieval. Here we demonstrated it on text and images, but it becomes super cool if we think about retrieving from electronic health records, retrieving from knowledge graphs, knowledge bases, and how do we build this type of multimodal models that have this ability. Maybe, you know, some kind of neurosymbolic way might be a way forward, but I think the trend in AI has been is, you know, kind of less inductive bias and more data. So if you have access to large scale data, then the best thing is just to train from it and learn the patterns from it, rather than trying to human impose them through some kind of inductive biases. Yeah, thank you for the question.
Jure, nice meeting you. Song. All right, nice seeing you again. And my question is, related to this RNA model, and have you thought about, for instance, integrating some of the foundation model genetic information and RNA, just the genome sequence, for instance? So in theory, all the information encoded in genome sequence, right? And then, of course, in different type of cells, they get expressed differently. How would you consider?
Yeah, but we don't know if we don't know why, right? Like that's exactly the key. Is like if you only look at the genome, then it's boring. It's one genetic information for me, but I'm composed of very different and very heterogeneous cells. So genetic information, genetic sequence is not enough, right? It doesn't describe me at the heterogeneity of my body, of my organs, of different tissue types, of different cell types in my body. So it's a much more faithful representation of a cell is not its DNA, but it's basically its RNA, because it says what kind of proteins are expressed or are present in a given cell, and it's really the proteins that how to say, control biological function and processes, right? So DNA, as the way we get it, of course, is important. But even in the DNA, right, you have all these kind of metagenomics when DNA is kind of wrapped in coils and so on. So there's so much that the DNA itself, like the genetic sequence, does not capture. So being at this RNA level describes the state of the cell much more faithfully.
Thank you. So actually, the part about his agent for research is also very interesting. And I wonder whether you have thought about, how it can be connected to some of these AutoML approach. So previously, when computer scientists, they try to tune this machine learning model, right? They use some kind of AutoML tool to search for hyperparameter space, or even for architectures and training data sets, features, and this type of thing. Could it be potentially be combined to make it more interesting, more powerful in some sense?
You mean for the research agent?
That's right.
Yeah, I mean, yeah, exactly. Like you could say this is AutoML, but it's even harder because I give you no code. There is nothing for you to start with, right? It's like textual description and a data set. It's what a human gets. Right, and usually when we talk about AutoML, it's usually, there is a set of hyperparameters, something to tune, something to play with, some search space. But here, the search space is the search space of all possible programs, right? So kind of the task is much harder, right? Because we basically just give you a data set and a textual description of the result we'd like to get. Definitely. So it's an interesting question how let's say AutoML could help. Maybe as a second step, but somebody has to write the initial architecture, has to, and that's also what's the part of the task.
That's right. Yeah. So actually related to this, are you thinking about building some kind of tools or packages to capture some of the traces for human researchers, the steps when where human researchers reason about the process, and sometimes this could be very good supervision to guide your agent to generate some intelligent behavior.
That's a super cool idea. We haven't thought of that, so that's not something we are doing or we are planning to. But I think that's a cool idea. If we could kind of learn not only from the end, but also like if we could fine tune almost like the model with this kind of human traces, that would be super cool. I think that's a great idea. We haven't thought so far. Thank you.
Okay, now let's have one question from the audience. Anyone?
Okay. Hi, Jure. This is Peng Cui from Tsinghua University. Long time no see. Very glad to see you here remotely. I'm very curious, interested in the third part of your work, just on the research assistant, of AI research assistant. You know that the mission of a researcher actually is to identify or discover some knowledge that is still unknown to the mankind, right? And the classical way for us to do this, I think, is just a hypothesis testing, right? And the hypothesis and the testing, I think the more important one is just how can we propose some valuable hypothesis. So I don't know whether in your system, in your AI research agent, do you think that the current large language model, they have the potential to propose some valuable hypothesis? I mean, in a quite, rational scope.
Okay, so what I didn't follow. What exactly is your question? Like,
I mean,
whether these agents would be able to discover something new?
Yeah, but ask some questions that is interesting, because you know that as a research work, the most important is idea, right? Of course, we can ask some questions to the large language model to find out the answer, to let it to help us to find out the answer. But I mean, the most important part is the question itself. So do you think that there is any potential for large language model to ask valuable questions?
I think that's very interesting. I think in this work with the AI research assistant, you could say that the model is trying to test hypothesis, that the assistant is trying to test hypothesis, is trying to find innovative hypothesis questions to solve a given task or to solve a given problem. At some level, but I think you are probably meaning this kind of more generally. It would be interesting, right? If we have, I think the first step to get there is to build this kind of closed loop systems, right? I gave you an example of AI research agent that could basically be a closed loop system that gets some task and then moves forward through a series of interactions. And I think to see whether the models have this ability to ask good questions and come up with good hypotheses would be that after we are able to do this on these well-specified data sets, give less specific instructions to these models and see, or to these agents, and see if they can come up with innovative solutions. So I think it's good, especially I think in sciences, in let's say biological, physical sciences. Having a good hypothesis is a starting point, and I think just kind of anecdotally, these models hallucinate so much that they come up with good hypotheses, I would say, or at least plausible sounding hypotheses. Whether they are kind of actionable or not is the next question.
Okay, thank you.
Thank you for the question.
Maybe I'll have one final question because it's very late. It can be either from the audience or on the present, or it can also be on the web. Okay. So.
Very nice meeting you again. This is Mu Qiao. My question is, do you think it's possible to align language of life, like encoding protein molecules with human natural language in a unified feature space?
And you would align them so that you are able to think of this as kind of as some kind of in terms of kind of multimodality and being able to mix the two. Is that
yeah, that's right. For example, projecting them into the same feature space through embedding all other techniques, then possibly we could ask natural language to investigate or explore those biological instances?
Yeah, I think it's very doable. I don't think why wouldn't it be? Right? Like so far we've been able to take many different modalities and kind of build multimodal models that basically embed or are able to reason across those modalities. So I don't see why let's say biology and natural language couldn't be. The question is more, is there a right data set that would allow you for large-scale self-supervised training of this, right? So at the end, I think that's really the question. I think whether it's doable or not from technical point of view, my answer would be that it is doable. The question is, do we have the right data set?
Okay, thank you.
Thank you.
Okay, thank you very much, Jure. Yeah, it's very for this wonderful talk and also the wonderful question and answering. So now let's give floor to Dr. Yang, and she will be leading from now.
Thank you so much for the wonderful talk. I believe that we all learned very much about AF science and AF biology. And I also want to introduce one of the big reading group that's started by your postdocs and your current students. They are Wu Tailing, Huang Qian, and Huang Kexin, and this reading group also just started like last week, and they're trying to promote your work and promote the works from this field. So thank you very much, Jure, and I know it's very, very late at your time, so have a good night, and thank you so much for coming to our conference.
Yeah, thank you so much, and have a great and successful rest of the conference, and appreciate the invitation. Good night.
Good night.